So yeah, this is year 4 of our project on how to change production practices of ornamental plants for better pest control. And that's interesting that there was a question on Barry's presentation, like what about the insects? So that's what this presentation will be all about. So first, I would like to introduce also all my the people that worked on this project. Definitely acknowledge the awesome contributions of Sarah and Siobhan from Omafra. And then there's a lot of violent technicians, some new faces as well. So we have Ashley, Laxmi, and Jessie, who did most of the experimental work. And then this year we were joined by the biochemistry team, Dr. David Liscombe and Kevin Houghton, who really contributed a new dimension to this project. So the original objectives were really to look at several things, and this is the text version, but I will show you a graphic that maybe represents it a little bit better. So we want to look at all the different things that you can do apart from doing biological control that will help round out an IPM program. So the red line in this graphic here represents a conventional scenario. So pests over the production time, pests like thrips will just increase in numbers if you don't do anything. And sometimes even with biological control, it's hard to get a handle on them. Now, the first objective was about dipping cuttings. Dipping cuttings was shown that you can reduce pest populations that way by killing them already at the beginning of the production cycle. So that's that a green arrow down. You start with lower pest numbers. And then the second part that we looked at is plant resilience. So how do we make the plant less nutritious or less attractive to a pest, which will also slow down pest populations, and then the biocontrol program will work better because it makes it easier for them to catch up with them. So one of the ways this could be done was by optimizing plant nutrition to benefit the plant, still get a good plant, but not provide a lot of excess nutrition to the pests. And as Barry explained, floriculture crops like mums are often over-fertilized. And all this research that Barry did, and also some people before him, showed that in different crops you can reduce nitrogen and other nutrients up to 70%, 75% without affecting plant quality. So this means there's actually room to play with different rates of fertilizer and see what happens with the pests. So our hypothesis was really that reduced fertilizer will have additional benefits, not only for your pocketbook and for the environment, but also for integrated pest management. And the way we think this works is that especially nitrogen in the form of proteins and amino acids is an important element for insect nutrition. They also need a balanced diet to grow very well. So this diagram kind of represents the plant. So at the bottom we see kind of the root zone. The middle is the leaves, and then at the top you see the shoots. And the arrows represent how nutrients are taken up and moved and stored in the different plant parts. So in general, the reduced fertilizer likely lowered the plant's nutritional value to pests, especially in the amino acids and the proteins. In addition, reduced fertilizer may also increase plant defenses, which is a very interesting option as well. So overall, our hypothesis was that it should slow down pest population increase and make the biocontrol program work better, or at least give it a good chance. So we looked at thrips and at whiteflies, and the thrips feed on the plant tissues and the whiteflies feed on the phloem sap, and both of these food sources have these amino acids and proteins. However, it may be possible that we would find that reducing nutrients too much would negatively impact plant quality. And this is where we were very interested to look at the role of biostimulants. Biostimulants such as beneficial bacteria, fungi, or extracts may mitigate these negative effects of reduced nutrition by helping the plants deal with nutrient uptake. And at the same time, as this graphic shows, beneficial microbes and other biostimulants can induce the plant to produce defense compounds or release volatiles that attract natural enemies, which also has a very beneficial effect on biological control and pest control in general. So putting it all together was quite a complex project, and I'll first do a little recap on what we found in previous years, and then I'll present what we found this year. So in the first 3 years, basically the dipping we finished up the dipping objective. So it's clear that dipping unrooted cuttings reduces pest populations such as thrips and increases the success of an IPM program. And we especially looked at thrips, but in previous projects, we also proved this for whiteflies as well. With regards to optimizing plant nutrition, we looked at 2 different cropping systems. We looked at whiteflies on Gerbera, and there we saw really that within the time frame of a potted Gerbera crop cycle, which is relatively fast, whitefly populations increase. They have a very long generation time, about a month even. And so within this time frame of a crop cycle, we did not see any meaningful effects of reducing plant nutrition to slow down whitefly populations, because they're already quite slow. Another possibility was that a less optimal diet for whiteflies would make them more susceptible to entomopathogens, such as fungal biopesticides. But in the graph here, you can see all the bars are basically at the same level, meaning that whiteflies at different nutrient levels were not differently susceptible to these biopesticides. And when we look at thrips on potted chrysanthemum, which was our other model crop and model pest, when you take all the results from all greenhouse trials together, we saw a possible effect of plant nutrition on thrips populations, possible reduction between 30 and 50%. However, there was a lot of variability. Some of the greenhouse trials gave us very different data than others. And we also saw that the low rate of that was meaningfully suppressing thrips populations was limiting to the plants, which you can see an example of in the picture here of the mums. So I would say there was potential, but we couldn't really yet formulate any recommendations for growers. Our first attempt to try biostimulants to make up for this negative effect on plants and to see if it maybe induced defenses against thrips also was not successful. Maybe because of the type of biostimulant that we tried, or some experimental effects. Now, when we combined reduced plant nutrition with thrips biocontrol, we saw no differences, and that's probably because we used a strong full thrips biocontrol program. So it was already so strong that we could not see subtle effects of plant nutrition on that. So like I said, we were not really ready to formulate any recommendations for commercial greenhouse practices. So we did a complete rethink of the project. We took into consideration several factors. For example, that the effects may depend on plant species or variety, the environment, the type of substrate used, the type of irrigation, the fertilizer type. So that gives us a lot of variability, probably. Also, what we didn't know was what happens in the plant. I mean, we varied nutrient rates, and then we looked at thrips at the other end, but anything that happened in between, we didn't know anything about. And of course, we only tried 2 different biostimulants, and there's so many biostimulants on the market that we figured that different biostimulants may work much better. So instead of doing more greenhouse trials, we went back to the lab and looked at the effect of plant nutrition on thrips biology on the much more controlled conditions and with different chrysanthemum varieties, not just using one variety. So here's the first experiment that we did. We looked at 4 different cultivars: Chesapeake, Grandview, Springdale, and Byron. And then we put them at 2 different, grew them at 2 different fertilizer rates in a growth chamber. And this was a slightly limiting under our conditions rate, 75 ppm of nitrogen, and a full rate 250 ppm of nitrogen. Then when the plants were grown enough, we took leaves from them and did choice trials with thrips and no choice trials on these detached leaves in the laboratory. So we took female thrips, we conditioned them on a leaf of a certain plant grown under high or low conditions. And then the next day, we put her on a new leaf and really looked at how many eggs she laid and how many of those eggs developed into first instar larvae, which was a very controlled experiment, much more than to really see if nutrient levels in the leaves had an effect on thrips oviposition and what this did in different cultivars. So I have to say this is still preliminary results because we've done it once. We have to do it at least one more time to really confirm the results. But what came out of it was already very exciting because we saw in the graph on the left that oviposition is somewhat affected by fertilizer levels, because on the y-axis you see the mean number of larvae that came out of the trial, and then the light colored bars are the low fertilizer plants, and the dark colored bars are the high fertilizer plants of the 4 different varieties. So differences were even more pronounced in the choice trials on the right. So the same kind of layout of the graph. And you see really thrips, if they had a choice between a low and a high fertilizer leaf, they all laid their eggs in the high fertilizer leaves. And what's also obvious is that there were large differences among chrysanthemum cultivars. So that's the thrip story. And hopefully our next round of experiments will actually confirm this. Now, what actually happened in the plant, we could see that through metabolite analysis. So we took leaf samples from the same plants that we did the thrips trials with and sent them to our biochemistry lab where they were analyzed with untargeted and targeted metabolite analysis. And what they gave us back is this kind of plots here. So how a plot like this works is the analysis looks at a lot of different concentrations of metabolites in the plant, hundreds of metabolites, and then it plots it out in 2 dimensions according to where the biggest differences were. So you can see directly that, hopefully this is not too small, but the different colors represent the different cultivars. So the cultivars were very, very different from each other with regards to their metabolites. And then within the same color, you have light and dark dots which are all mixed together. So you can see that there were very few differences due to fertilizer rate. So we did it once, which is the graph on the left, and then on the right we did it again, and very similar results were obtained as well. So that means that the effects due to cultivar were much greater than the differences due to fertilizer rates. There were a lot of metabolites that were changed, but one of them was very interesting and varied with high and low fertilizer, which was chlorogenic acid, which is a known defense chemical in chrysanthemums. These two graphs represent the 2 times we did this, and especially in the graph on the right, you can see that the low fertilizer plants had higher levels of chlorogenic acid than the high fertilizer plants. So there was more resistance chemicals in the low when you reduced fertilizer rate. And there were other metabolites that also varied with fertilizer rate in all cultivars. And who knows, maybe there's other defense chemicals there as well that we don't know yet about. But the identity of these compounds, we still have to see what they are. Now, the next step is, of course, to repeat this, but an experiment that's ongoing now is to look at the effect of different biostimulants. So this time we chose 4 biostimulants, some that are not yet on the market, like Bacillus pumilus and a strain of Bacillus subtilis, and then we compare it to Root Shield Plus and actinovate, which are known commercial products. And of course a control, and we applied them at 3 different times in the production cycle, at 2 different fertilizer rates, the same as before, and at 2 different cultivars, because we've seen that these cultivars are so different from each other. So we're going to look at plant growth parameters to see if biostimulants can make up a bit for the reduction in nutrients available to plants. We'll also look again at thrips overposition in the no-choice trials. We'll do metabolite analysis and SAP analysis, a thrips damage trial, and a shelf life assessment at the end to see if biostimulants help plants with water stress. So a whole host of different measurements in one experiment, and that experiment is going on right now. So in conclusion, we can say that lowering fertilizer rate may reduce losses due to thrips. There are some indications already that there are some effects. But selection of resistant cultivars is one of the main important things for thrips IPM probably in chrysanthemums, because the differences between the different cultivars were so big. And then if you do grow susceptible cultivars, you can concentrate your bioprogram on the most susceptible plants and maybe even use them as trap plants. And this is all in addition to the benefits you get, as Barry explained, by reducing fertilizer application that will lead to cost savings through reduced inputs. So in the last year, we'll be continuing these lab trials in combination with metabolomics. I think we have a very good thing going on. Combining these 2 things, we'll get a lot of insights in what happens in the effect of cultivar, also see what the effect of the biostimulants are. And I would still like to try and do some commercial greenhouse trials. What exactly we're going to test still has to be determined, but I think there are some things we can test as well. And then lastly, we gave quite a few presentations also in for COHA in French and in English. There was a poster at the Canadian Greenhouse Conference, and there will be a letter in newsletter coming out soon as well. So with that, I'd like to thank the whole biocontrol lab and all the staff at the Violent Greenhouse, Research Greenhouse, the statistical analysis, as well as our commercial partners and the funding from COHA and the Government of Canada. Thank you so much.